early 60s, Denham Harmon at the University of Nebraska began to formulate a theory of free radical disease. And he had particular emphasis on cancer and aging. Now, free radicals are chemical species that are characterized by an odd number of orbital electrons or by pairs of electrons of similar directional spin isolated singly in separate orbitals. Pro-oxidants, reactive oxygen species, and free radicals are often considered as synonymous terms. Consequently, most of these agents are highly reactive and usually exhibit an extremely short half-life and are capable of doing extreme damage to tissues. In the free radical hypothesis, Harmon stated if free radical reactions contribute to the degradation of biological systems, and if the level of deleterious reactions can be lowered significantly by diet, then such dietary alteration should have a beneficiary effect. And he made three suggestions or recommendations. The first was caloric reduction, that is, lowering the levels of free radical reactions arising in the course of normal metabolism. The idea here is that you consume higher levels of calories, then you're stoking up the fuel, and consequently you're getting an increase in loss of electrons that ultimately end up in reactive oxygen species. Now, the second one was to minimize dietary components, for example, polyunsaturated fats that tend to increase the level of free radical reactions. And the third was to supplement the diet with one or more free radical reaction inhibitors, that is, antioxidants. Now, what I'd like to do today, this afternoon, because the title, if you remember, was refinement of some of these dietary recommendations, is to look at the latter two, that is, minimize dietary components such as polyunsaturated fatty acids and supplementation of the diet from one or more free radical reaction inhibitors. Now, in terms of minimizing dietary components that tend to increase free radical reactions, this is the kind of reaction that Harmon was talking about, where you have a radical attack upon a polyunsaturated fatty acid, and these fatty acids are particularly vulnerable to radical attack. You get a polyunsaturated fatty acid radical, and in the presence of oxygen, that radical will produce a peroxyl radical, and by hydrogen abstraction from the adjacent polyunsaturated fatty acid, you get an autocatalytic reaction leading to another polyunsaturated fatty acid radical and a hydroxy peroxide. Now, in relation to the effect of dietary polyunsaturated fats to UV carcinogenesis, the areas that I've worked in, we have supplied various levels of dietary fat to animals and then induced skin cancer in these. And you can see that basically you get almost a linear response with respect to increasing dietary lipid level in the diet. Okay, this is in terms of tumor latency. If you throw in an antioxidant or a cocktail of antioxidants, you can see at the highest dietary lipid level you get a significant inhibition. However, as you reduce the dietary lipid level, of course you're getting less tumor, then basically the antioxidant effect begins to wash out, and at the lowest level there is no effect. It's completely gone. Well, this looked as though basically what we were doing is just supporting what Harmon had stated. As you add polyunsaturated fatty acids, you get more lipid peroxidation and so on. But on closer inspection, what we found was that in the cutaneous tissue, the skin, basically antioxidants were inhibiting the stable lipid peroxidation products, NDA or TBA, reacting substances. And if you look at the various levels of dietary fat, you can see that basically you have inhibited, that's this bar right here, in all the different lipid levels, lipid peroxidation products to about the same level, while you still have an increase in carcinogenesis. So obviously there's something else going on here than just simply lipid peroxidation. As you know, there are two series of essential polyunsaturated fatty acids. The first, of course, and the ones that we use in the studies to show or demonstrate the effect of dietary fat on carcinogenesis, were omega-6 fatty acids here. And basically, linoleic acid was the main source of omega-6 in the diets that we supply the animals. The other class are omega-3 fatty acids. And they are metabolized through a cyclooxygenase and lipoxygenase pathways. And what happens here is that you get differential products as these two types of fatty acids will flow through the cyclooxygenase pathway. Here you see a major pro-inflammatory and also tumor-promoting compound is PGE2. Whereas with the omega-3, you reduce the levels of PGE2 and you get PGE3, which is less inflammatory and is not a tumor promoter. And basically what we were measuring when we were looking at lipid peroxidation was the final flow of lipid peroxidation through these pathways. Now, omega-3 fatty acids are actually, they have a higher iodine number than omega-6. So that means that they are just as polyunsaturated, even maybe a little more so. They behave that way. You would think then that there are a number of immunomodulators that are produced in this pathway, the prostaglandins and so forth. And I think you saw this slide perhaps the other day, but if you have a low dietary fat level as compared to a high dietary fat level, you get immunosuppression with this high fat. And of course, this means that you're producing more immunosuppressing materials through the cyclooxygenase pathway. This is then the kind of effect that you see, a comparison of effects of omega-6 and omega-3 fatty acids on UV carcinogenesis. You can see with the omega-6, this happens to be tumor latent period in weeks, and you get a much longer tumor latent period with omega-3 fatty acids. This is tumor multiplicity, that is numbers of tumors per animal. You can see with the omega-6 as compared to the omega-3, you get a very pronounced inhibition of carcinogenesis. Now, if you compare these, then experimentally, dietary 6-omega fatty acids exacerbate UV carcinogenesis, dietary 3 inhibits UV carcinogenic expression, dietary omega-6 exerts its principal effect on the post-initiation or promotion stage of carcinogenesis, and the influence of omega-3 fatty acid on carcinogenic expression is exerted across the carcinogenic continuum, that is, an initiation and promotion. Pro-inflammatory and immunosuppressive PGE2 levels are increased linearly as omega-6 fatty acid concentrations increase in the diet. Pro-inflammatory and immunosuppressive PGE2 levels are dramatically reduced by omega-3 fatty acids. Omega-6 suppresses the immunologic responses involved in tumor transplant rejection, and the immunologic pathways manifested in delayed type hypersensitivity and also in contact hypersensitivity. Let's see if I can go back here. And dietary omega-3 fatty acids inhibits UVR suppression of contact and delayed type hypersensitive responses. So in regard to this tenet 2, that Harmon suggested, a general indictment of polyunsaturated fatty acids with respect to lipid peroxidation no longer is a reasonable tenet. The response to dietary polyunsaturated fatty acid is much more complex and has to be approached on an individual polyunsaturated fatty acid basis. Now, the second tenet that I wanted to talk about was the potential antiradical strategies in cancer prevention, that is, a supplement of the diet with one or more free radical reaction inhibitors. And the idea here is that, remember, we had a fatty acid peroxyl radical, and if we then have an antioxidant, it is going to protect this polyunsaturated fatty acid, and we're going to synthesize a polyunsaturated fatty acid hydroxyl. We're going to terminate then that autocatalytic reaction. And I think you might have seen this yesterday, but this is the effect of one antioxidant. It happens to be butylated hydroxytoluene on UV carcinogenesis. These animals were not supplemented. These animals were supplemented in their diet with this antioxidant. So we know that we can protect then against UV carcinogenesis with some antioxidants. But I would have to say that you have to remember that under certain conditions, an antioxidant may act as a prooxidant. For example, beta-carotene, and I'll talk about in a moment, under low oxygen tension, it is a very effective antioxidant. However, under high oxygen tension, it behaves as an oxidant. So you have to remember these, that under any kind of condition, the antioxidant you use may behave quite differently under some conditions. And their actions are very specific in specific tissues. Well, one thing about BHT is that it's been reported to act as a tumor promoter under certain protocols. And so it's probably not a good candidate, basically, to use in human diets, although we do consume quite a bit of it. BHT induces hepatomegalia and hepatic phase 1 and 2 microsomal detoxification enzymes. And this kind of, even though we saw no pathology associated with hepatomegalia, basically it was related to the induction of these enzymes, which means that when we took these enzymes and isolated them from the liver of animals that had been fed the antioxidant, and then tested them with a very potent hepatic carcinogen into FAA, basically you can see that what happens is that you've got a mutation frequency here for the control, that is non-antioxidant supplement, and here you've got a very marked increase in activation of the carcinogen with the antioxidant supplement. So basically what you may be doing then, this antioxidant may be acting as a double-edged sword in that it does protect against UV carcinogenesis, but it predisposes perhaps the host to some other type of carcinogenesis. The second example I wanted to talk about was beta-carotene. A study in 1981 by Pito and colleagues found that human cancer risk, and this is an epidemiologic study, were inversely correlated with dietary beta-carotene, okay? Blood levels of dietary beta-carotene. Well, this spawned, or helped spawn, a international multi-billion-dollar, about $40 billion industry in food supplements, of which a large part of this was antioxidants. Now, this industry, because many of the antioxidants like beta-carotene are natural products, vitamin C, vitamin E, and so forth, the industry is not very well regulated, and consequently, long-term tests for detrimental effects of these supplements are not provided. In our hands, with UV carcinogenesis, you can see and we were using a semi-defined diet, that under certain dietary conditions, beta-carotene can actually exacerbate carcinogenesis. This is with respect to tumor latent period, a shorter latent period, and also in terms of multiplicity, you can see more tumors per animal with the beta-carotene supplemented diet. Furthermore, in persons with a previous non-melanoma skin cancer, treatment with beta-carotene didn't reduce the occurrence of new skin cancers over a 5-year period of treatment and observation. And the real hooker in this whole thing was this study by the alpha-tocopherol beta-carotene cancer prevention study, in which they provided beta-carotene supplements for 5 to 8 years to male smokers. And the outcome of this was that there was an increase of 18 to 19% of lung cancer with the beta supplemented group here. So there are other adverse effects of antioxidant supplementation. It really depends on how you want to handle the data, I suppose. But the first study that was published in the Journal of American Medical Association in 2007 was a meta-analysis that concluded that vitamins A, E, and beta-carotene may increase mortality risk by up to 16%. Okay. And it depends on cherry picking the data, but another reanalysis reported that you got a beneficial effect of about 42%. So we know that antioxidants prevent health promoting effects of physical exercise in humans, and there are a number of other adverse effects. So this has led the IARC to conclude that until further insight is gained, beta-carotene shouldn't be recommended for use in cancer prevention in the general population, and it should be assumed that beta-carotene, or should not be assumed, is responsible for the cancer-protecting effects of diets rich in carotenoid-containing fruits and vegetables. And then the World Health Research Fund and the American Institute for Cancer Research has stated that a general recommendation to consume supplements for cancer prevention might have unexpected adverse effects, and thus dietary supplements are not recommended for cancer prevention. And so the conclusion is that if you are going to use dietary supplementation as a means to reduce cancer risk, this is a course of disease prevention not without risk, and should be approached very cautiously. And I'm going to skip over this slide and go to the final one. The best recommendation for maintaining a strong defensive antioxidant system may be the consumption of a balanced diet containing adequate green leafy and yellow vegetables that are known to be rich in a broad range of antioxidants, and from which the original observations of reduced cancer risk were based. Thank you very much.
So you are not going to recommend taking any vitamin supplement?
Well, I think if you're consuming a